I have Alistair McTaggart, who has been collecting specimens and leading Australian research on this intriguing mushroom, admired by foragers and observers alike. His research has led to a new appreciation of the diversity of Psilocybe cubensis in Australia and the possibilities that this species may hold going forward.
Hey, Alistair here, and if you don't know me, I've been working on magic mushrooms for almost getting up to 3 years now. 2.5 years was pure research at the University of Queensland, and then the last 6 months has been such a blast because I've been working at a magic mushroom farm, starting to kind of translate some of the things that I was researching. Pretty groused. Today, Ronnie has asked me to take you through one of the pieces, but you're going to get two, that we published on Psilocybe cubensis. And I'm going to take you through it as if I was reading a scientific paper. So we're going to look at all the figures and kind of get to really understand what we did. And then it'd be crazy not to look at also the work that we've done on Psilocybe subaeruginosa. And it's so cool to compare cubes to subs here because you get to see what's happened under domestication or naturalization compared to what happens in the center of origin. Really easy to get your hands on the current sub piece. It's under review, so we've just made it publicly available. But the cube piece, it's behind a paywall. You might have to pay for it, so please do not pay for it. Contact any one of these co-authors here or me, and we'll send you a PDF with a big smile on our dial. If you wanted to, you could look at our original piece, which is called The Manure Tour. You just have to Google The Manure Tour, and you can kind of see what we were originally thinking to frame the cube work. After it got reviewed, it was kind of changed for a more scientific audience. I'd like to thank all the usual crowd, my collaborators and co-authors and colleagues who've helped me along this journey. Tanner and Otto join this list of thank yous. Thanks guys for sending me so many cool images. I'd like to give a shout out to Andy B here because to be a co-author on this work, you just had to send me a spore print, and somehow Andy B, who's the very first person to ever send me something, a spore print, he got left off due to a communication error by myself. So big apology to Andy B. Hopefully I can make it up to you in the future. I'll just quickly go through what we did in each of the two pieces so that you're on the same page and so that it's in the back of your mind when we're looking at all of these figures. So the cube work, ultimately what we've shown is that everything that's under cultivation has gone through a process of domestication, and that domestication came from a low founding diversity, about 7 populations, and I'll talk about why it's 7 or more coming up when we talk about mating loci. Because these low founding populations have been under domestication for the last 50 years, they've wound up with really homozygous genomes and they've lost allelic diversity over time, and then they've been supplemented by new collections from the wild, which is really, really cool. The population of cubes in Australia is 99.9%. We can be pretty sure it's been introduced to Australia, not a native species here. And because it hasn't been under domestication, it's just been randomly outbreeding and living the dream in manure. It has allelic diversity that is not present under domestication or in the cultivars, and it's going to be so cool to start mixing in some of the genetic diversity from naturalized populations of cubes in Australia with the cultivars. So directly comparing cubes to subs is really interesting because you get to see the allelic diversity that's been tinkered with or that's not in a natural population, and then you come along to subs and in the center of origin and you just see, wow, this is what allelic diversity should look like in a normal population, in a wild population. So Australia has super high allelic diversity at things like mating genes or psilocybin genes. We've got these geographically isolated populations, and sometimes on mountaintops or in valleys, and they're technically sexually compatible. There's evidence of past gene flow, but not really contemporary gene flow. So we've kind of recommended, well, we should still call them just all Psilocybe subaeruginosa, and that goes for the same species in the Northern Hemisphere, things like Psilocybe cyanescens or Psilocybe azurescens. They are pretty much Psilocybe subaeruginosa, they're populations or subpopulations of subs, and they could be safely called subs. And then the last thing I'm going to talk about is how cool the diversity, the allelic diversity of psilocybin is in Psilocybe subaeruginosa, and maybe some of the evolutionary processes that are driving that diversity in the wild. Okay, so this is in the Cube paper, and this is the first figure and also a supplementary figure. And really what we're seeing here, we're seeing these two SplitsTrees, which entheogenesis viewers are so familiar with now because I've shown them so many times. Essentially, it's just a way to visualize relationships among, in this case, genomes of Psilocybe cubensis, 124 genomes. And what you'll see is the Australian blue population is really different. It's like on this long branch compared to all these cultivated or cultivars, so the cultivated population. And a SplitsTree branch length is indicative of genetic distance, and reticulation explains complex evolutionary events such as sexual reproduction or recombination. So when things are reticulate, something was probably happening between them. So yeah, SplitsTrees you're putting down every single relationship there is among the data. Now, where I would actually start looking at this figure is technically figure C here, which is a structure plot or from k-means clustering, and it's how the colours were defined in these groups. So I've taken entheogenesis viewers through this before, but this is a way that we can potentially add groups to see how many groups are in our data set. And this is based on SNP data. This whole figure on the left is based on SNP data, and this is based on genes, this SplitsTree on the right. But yeah, each horizontal row we're adding a hypothetical group, and each vertical row is an individual. So if we had on here k equals 1, everything would be one population, and it would all be one colour. So we start at k equals 2, and we have this clear Australia is different to everything under cultivation. And that's kind of reflected in our SplitsTrees, where Australia is totally different to everything under cultivation. And in this two-dimensional discriminant analyses of principal components, where Australia is separated along this x-axis from the cultivated populations. So as we add more populations, we get to about 6 populations, and then nothing really changes when we keep adding more groups to these data or to this data set. And so that's where the colours come from, is this k-means clustering. And then we can see in this 2D plot is a way to visualise how genomes cluster based on that same data, so whether they're close to each other or not. And you can see that there's a little bit of ambiguity. So even though everything here is made a green colour, we've got something like, I think this is Escondido, Mexico, and it clusters really close to this green group, which is the luminous group. And I just want to talk a little bit about it. To me, it's one of the most exciting kind of populations. We've called them populations. They're not really populations because they're contrived and they've been outcrossed or they've been under cultivation, and things have changed as people have crossed them. So they're not true populations, but we've kind of used it as a group based on k-means clustering. And so this green group, luminous, is really an eclectic mix, and most likely it's taken from multiple different provenances and it's just being clustered together. And you've got something like Chitwan here. I remember when I first started analysing Chitwan, I really thought that there was a whole bunch of genome contamination because it was so different to everything else. But really, it's just probably something that's been collected recently and added to the genotypes under cultivation. So super cool. And in luminous, you kind of have examples of that happening. So you've got these colours. They're not true populations. They're not neat groups because we've had stuff going on under domestication, and the SplitsTree kind of shows that when you've got purple or pink mixed together and things that are super close, but they might have different colours. And that's probably in the process of breeding or in domestication, things are getting crossed. This is an analysis of kinship, and you can think about any sort of relationship here, any familial relationship, a brother and a sister or brothers and sisters, they share about 25% of alleles with each other. That's just based on the random inheritance or segregation from parents. And so in this plot of kinship, I knew which basidiospores came from the same parent, and I was able to start to apply whether things were kin. And we can plot it out. This is a pairwise plot, and as something is a darker red, it has more chance of being a kin relationship with that pairwise comparison. And then there's just a cutoff based on the Australian kinship. And we kind of are looking for these triangles along this, or that you can see sort of triangles in this big plot. So Australia is this big triangle, and then you've got these little kind of sharing genotypes or sharing alleles, probably more than 25% of alleles in a whole bunch of these. So they probably came from the same pileus. So Australia has some evidence of kinship. Then you've got the kind of whole cultivated population shares some level of kinship. You've got a darker triangle in here, which is your Malabar, Golden Teacher, and Blue Magnolia groups, in inverted commas, and then you've got your Luminous group and Penis Envy up here. So some level of kinship. Now, whether that's true or not is debatable. There's probably a level of kinship based on outbreeding or outcrossing or people trying to hybridize these different cultivars, and that can potentially cloud the issue here, but it's pretty clear that Blue Magnolia, Malabar, and Golden Teacher, the individuals in those groups, probably share some level of kinship, and Australia probably shares some level of kinship. Something like Chitwan, which I was just talking about before, you can just see it's kind of like got a really lighter line just through that triangle there. So Chitwan's a bit different. Okay, this is the hardest figure to understand in the manuscript or in the paper now, and that's just because there's so much going on. So figure A is an analysis of homozygosity and heterozygosity in individuals, and it doesn't include the Australian data because I wasn't looking at dikaryotic individuals, I was looking at haploids. So this is just in the cultivars or in the cultivated population. And so we're measuring something called the inbreeding coefficient, and in a normal population, you kind of expect things to be about 0, which means you have an expected number of heterozygous or homozygous sites. As you depart from 0, so as you get closer to 1, you have a tendency to have more homozygous sites. And this is what's happened in Penis Envy up here, like a really tight little group, or Golden Teacher, where you've just got, in these box and whisker plots, you've just got all your individuals approaching 1. So they tend to be more homozygous genomes, which is usually a sign of inbreeding. Something like Luminous, where you've got this long box and whisker plot, you've got probably up here where you've sort of departing from a 0, so you're getting closer to 1 and have more homozygous genomes. This is probably a cultivar like Luminous, which has been under cultivation for a while. And then at 0, and even in negative values, you've got individuals. Negative values would be probably an indication that you're having a whole bunch more heterozygous sites than expected compared to the rest of the population. So that could either happen because we're looking at a contrived population and everything is so homozygous, or it could potentially happen because you're crossing homozygous genomes from different provenance and everything ends up being heterozygous in that remaining cultivar. So that's pretty cool. Probably the message here is that Penis Envy and Golden Teacher are super homozygous genomes in general. This analysis, which you've seen before, is we're using something called linkage disequilibrium, which you could think of it as how random your genomes are along a chromosome, and do you have random SNPs, or do you end up having SNPs that are always the same no matter how far apart they are based on distance in the genome. Okay, yeah, that's under-explaining it, but it's a hard concept to understand. And so we can model something called effective population size from this. And the thing that I really want to draw attention to, and what excited me so much when I first saw it, this is kind of the evidence to me about why Australia is likely an introduced population here. and that's our effective population size has this massive drop, and then a recovery, which would be the equivalent of a couple of individuals arriving in Australia and then outbreeding again, and getting up to that effective population size after a massive decrease in effective population size. The cultivars all tend to decrease in their effective population size in recent generations. So potentially they've been only under cultivation for 50 years, and Australia is probably introduced here sometime in the last 200 years, just to give you an idea of generations there, though it's really hard to know what that actually means because of people keeping individuals alive clonally. So that can really interfere with this kind of generational estimations as well. When you have, immortal genotypes. So these next three figures are measures of genetic diversity, and probably the ones that are most interesting, well, the one that's most interesting to me is this nucleotide diversity, and it's a measure literally of nucleotide diversity, how diverse your genomes are. And what is telling is penis envy and golden teacher across the 13 chromosomes of Psilocybe cubensis. When you measure each chromosome for nucleotide diversity in those populations, you have low diversity compared to Australia and the luminous populations. And so we're really getting this message clearly that Australia and luminous are a little bit different to everything that's under cultivation. And that's because luminous has probably been supplemented with individuals from wild populations or naturalized populations in recent times. And Australia, as we know, is an introduced population. Tagemas D is a way we can measure the type of evolution or whether there's some sort of selection occurring. And in this case, we probably didn't do this analysis. We sort of did this at a high level. If we were really trying to drill down to sites that were under positive selection, we probably want to zoom in on suspect sites and have a look at those. But we're looking across entire chromosomes here, and what we're specifically looking for are sites that suddenly maybe decrease in their Tagemas D, which would be maybe a sign of positive selection or a sign of selection from domestication. And this is a site where it ends up in the entire population. Every individual ends up having this particular genotype or gene or allele or whatever, and that's because people have selected for it. At our scale, which has these long window sizes, it's probably not fine enough to resolve these individual sites. I did an analysis of positive selection, but again, I think my pipeline probably wasn't fine-tuned enough to pick out individual genes that were under positive selection. And I think this will be an exciting little kind of question if somebody ever gets into it in the future, to look at if domestication has had an impact on individual alleles that might be under positive selection. This is one of my favourite figures. The reviewer hated it, so it ended up in the supplementary figure, and it's kind of repeating that analysis of the inbreeding coefficient, or not repeating it at all, sorry, it's just another way to visualise homozygosity and heterozygosity. I've plotted homozygosity and heterozygosity as density across the 13 chromosomes of Psilocybe cubensis, and the big blocks of colour is density of homozygous sites, and the kind of spikes in front are the density of heterozygous sites. And so you can see over penis envy, and what I plotted was the two most and the one least heterozygous genome from each population or each group. And with penis envy, you've got one that's a little bit heterozygous, that's one of those Erisa genomes, and then true penis envy has these massively homozygous genomes, and you expect in a tetrapolar mushroom, so something that's dikaryotic, it has two nuclei, one from each parent. Those nuclei can only be together if they differ at two different parts of the genome that control mating compatibility, and that's on chromosome 1 and chromosome 10. And so really it's sort of like in penis envy, that's almost the only sites that are heterozygous, and the genomes are largely homozygous. Other than that, that other homozygous group was golden teacher, and you can kind of see that the same heterozygous pattern is almost repeated across these three genomes, so the two most heterozygous and one most homozygous genome. And it might be an indication that the diversity maintained in the sites is just present in all of them. So there's probably some sort of random mating going on under cultivation, but there's not a lot of diversity around. And then something like Chitwan here, this is this top green one, and that's what a true mushroom, probably plucked from the wild, or maybe in the center of origin or in a naturalized population. That's the kind of heterozygosity that we would expect if we got and had an opportunity to do this for all the Australian ones as well. So I quite like this figure, and it's just a way to visualize who's really homozygous and what normal heterozygosity might look like. Okay, this is the last little analysis for the cubes, and it's about the mating compatibility loci. And this is just one of the genes behind whether fungi can cross or not. So whether two nuclei can go into the same piece of hyphae. And what we've done here is just pull out all of these mating compatibility genes and put them in a phylogenetic tree. And then this middle thing is a bubble plot that kind of shows how many of that allele occur in the populations, and then what they occur with. So the three most common alleles are A, B, and C, and they're really prevalent in golden teacher, blue magnolia. And then you start to have these smatterings of other mating compatibility alleles come in. So something like Malabar has something a little bit different, has a different allele. And then you've got the Thailand provenance ones, and then Luminous, just scattered around the tree. And so Tim James and I were talking about what this means in a normal, in the center of origin, in a natural population of mushrooms. These mating loci end up accumulating huge amounts of allelic diversity over time and space. And that's sort of, it's not because of tetrapolar mating, but it's as you, if you need to cross with something that differs at both that differs at two sites of the genome, you end up kind of having a lot of genetic diversity or allelic diversity at these sites that control compatibility. So you do expect there to be large amounts of allelic diversity at this particular gene. In the cultivated populations, you can clearly see there isn't, because there is only really 3 dominant alleles. And we get new alleles from Luminous and Thailand and Malabar. So there's 14 mating type alleles in this tree, and Tim and I were saying this is probably an indication there's at least 7 provenances of Psilocybe cubensis under cultivation. So you would get to that because you've got 14 different alleles, but because they have to be heterozygous, that kind of translates, you divide it by 2 to 7. So all the mushrooms under cultivation included in this study came from at least 7 provenances, and that's what's under cultivation. So it's actually not a lot. If we went to the same sites that I have in Australia and we saw, I haven't counted how many HD alleles are in the Australian population, but it's comparable. So Australia probably has more allelic diversity at these genes. Okay, on to subs. So really a parallel analyses that are happening here, so we can kind of see immediately the difference between cubes and subs, and you've had practice looking at it. So we've got genes and SNPs on the left here, and we'll start with our little k-means clustering group, where we've defined the k-means clustering to define the groups. So at k equals 1, it would all be one population. At k equals 2, we get this split of red and blue, and we see it again reflected in the other measures of this genetic diversity. So our red group in our 2-dimensional plot, in our DAPC, is away from everything else, and then in our splits trees we've got this kind of red group compared to everything else, and that the red group is everything south of the Great Dividing Range, so Tassie, and west of the Great Dividing Range, so central Victoria and South Australia. And then as we go down, we end up with 6 again, and it's sort of reflective of the sites that were sampled, the number of sites that were sampled. And where I've been trying to hopefully take you to my way of thinking is that each of these clusters that you see in the SplitsTree or in the DAPC, each of those is one dikaryotic genotype. So we had close to 90 individuals sequenced for this study, but the reality is we only have 1, 2, 3, 4, 5, I think there's about 20 different groups. So all of our individuals probably only came from about 20 different dikaryotic mycelia. If we kept sampling from dikaryotic mycelia all across Australia, we would gradually expand out these SplitsTrees to be big wagon wheels that all reticulated in the middle. The reason that I think this is because something like Ravensbourne, which I know all came from one Pileus, all clusters together. So we can kind of see, well, this is what siblings look like. We go have a look at Bunya, which came from 3 different mushrooms, A, B, and C, and they all cluster together. Kane and Bill sampled from Clifton Hill and Geelong, 2 disjunct sites. Kane kept his pilei separate, so we can see E there. He collected from one mushroom, Bill collected from many mushrooms, but they have a sibling relationship, and it's probably an example of a council or a landscaping company spreading mulch all around Victoria and spreading that one dikaryotic mycelium of Psilocybe subaeruginosa. In Tasmania, we've got Jan collecting in Ellendale. She collected 3 different mushrooms or pilei and made spore prints, M and N and O maybe, sorry. And she's got these 2, she collected those 3 from 2 different dikaryotic mycelia. And the same from Kane in Kununyi. He's collected 3 mushrooms again from 2 different dikaryotic mycelia. So that's the way to think about subs. There's this persistent mycelia that exists over time and space in natural areas of Australia in the wild, and it's just fruiting out mushrooms every year. So you probably are going back to the same genotype over time. I expect that we'll find, if we just kept this study going, we would eventually find the dikaryotic mycelia for Psilocybe cyanescens or Psilocybe azurescens, so something that maybe could be linked to being a parent of the founding events for the mushrooms in the Northern Hemisphere. So the kinship plot, similar to our study on cubes, we've got this pairwise analysis. We've got very strong support for siblings. Maybe even I've set the cutoff too high. And we've got these 2 big triangles. So a big triangle here, which is kind of that east and northerly populations, and a big triangle here, which is that west and Tasmania populations. And then there's also some evidence of kinship with northern Victoria and southern Victoria, and that kind of shows that there is kinship, or there is some sort of past gene flow going on, or some similarities at some point in time among all these different populations. And because of that, so because we know that they're very, very closely related, these are populations that are scattered all over Australia, and even if they have very little chance of contemporary gene flow, do we still call them the same species? And this is a phylogram, a phylogenetic tree with informative branch lengths of the ITS region here on the left, and then these are haplotype networks. And in each haplotype network, I've taken one of the genotypes of ITS here and fully, from GenBank or from wherever from this data set, showed how many individuals share that genotype. So in something like this one, we've got 34 individuals share this one genotype of ITS, and it differs to this one by 1 parsimony informative character. So the number of dashes or hashes on each line is the number of parsimony informative characters that are different between those 2 genotypes of the ITS region. So Ultimately, what it means is that there's heaps of different ITS copies around Australia in our natural populations of Psilocybe subaeruginosa, and it includes ITS copies of those that have been sampled in the Northern Hemisphere from things like Psilocybe cyanescens and Psilocybe azurescens. So really they are all the same species, even under hypothesis that ITS is a good measure of biodiversity in these individuals or in this taxon. The two that do stand out are these two from New Zealand, so Psilocybe weroroa and Psilocybe, macarorae. And I think for Psilocybe macarorae up here, it is quite different in the ITS region. And what ultimately will need to happen is that people in New Zealand will need to sample populations of these different taxa, if they think that they're different taxa, and add it to the sub-data to see how they're related to the sub-populations. Are they going to be distinct species, in which case, based on ITS, Psilocybe subaeruginosa would be paraphyletic to the New Zealand taxa, or are they just subpopulations of Psilocybe subaeruginosa? So are they just the New Zealand populations? Now, I have said this before on my blog, taxonomy to me, when a name has been validly described, you can use it forever. Taxonomy is about communication, and I would never say to not use a name. Please keep using Psilocybe cyanescens. Please keep using Psilocybe azurescens. These are informative of populations of Psilocybe subaeruginosa in the Northern Hemisphere. And in terms of communication, as long as you understand that Australia is the centre of origin and Psilocybe cyanescens is something that has escaped from here and gone into the Northern Hemisphere, then I'm cool with that. So use names for that have been validly described for sure. In cubes, we sort of see this same diversity or some genetic diversity at the ITS region. So here's everything from Australia in 2 different genotypes. Most of the cultivars have 1 genotype of ITS, which we would expect because they have really low genetic diversity. But yeah, the point is that we do expect variation in the ITS region. And now if you're saying, man, Alistair, you're not recognizing all this phenotypic diversity, you jerk, you're decreasing biodiversity. Oh man, I'm sad too, come on. Because when you look at all the phenotypic variation among Psilocybes in Psilocybe subaeruginosa, you're like, wow, it's so variable. You've got these beautiful caramel coloured caps or these chocolate brown caps. You've got sinuate pili, you've got nice conical pili. There's a lot of phenotypic variation there, and it might seem like you're bummed out by having fewer taxa to represent this kind of natural phenotypic variation. But the reality is, based on genes and gene flow and everything that we've just seen up until this point, the genetics are saying, hey, this is one species. With cubes, I'd just like to point out there's a lot of phenotypic variation in cubes as well, especially since I've been growing them and starting to see sinuate caps and whatnot. So you've got, your flat and your really rounded caps on Psilocybe cubensis. I've seen sinuate caps, like I just said, and then this is treasure coast here. I don't even know how you would describe that morphology. So it's rich with phenotypic variation as well, but we still just think of cubes all as cubes. So subs and cubes, both one species. And what you see as phenotypic variation is what we would expect. Just the same as humans or every other living thing has phenotypic variation. It's what makes us interesting. Okay, this is a comparable thing to that mating compatibility study in cubes. So what we see here is, remember, if I've only sampled from 20 dikaryotic individuals, the maximum number of alleles that we could have is 40. And that would mean that each population has 2 different alleles at their mating compatibility loci. And that's pretty much what we get here. So really high allelic diversity at the mating compatibility genes. The lines that are connecting things, that's from my sexual compatibility crosses. So I'm crossing haploid hyphae from different locations to see if they're compatible or incompatible. And that's just based on clamp connections rather than formation of a mushroom. Because at that point in time, I didn't have a permit that let me fruit mushrooms. I could only cross hyphae. And probably the most telling cross is the Bunya Mountains way up in Queensland. So the most northern population sampled crossed with populations from Tasmania. So absolutely these geographically distant populations are still sexually compatible. And that's based on their mating genes. So they're very different mating genes and they still cross. Well, I shouldn't say very different, but yeah. Okay, this is the good stuff. So everything now is about psilocybin loci and what I've been doing with them lately. So in cubes, here's that haplotype network again. You're familiar with them now. And what we pretty much see, so each of these big circles is one allele of psilocybin, the 4 to 5 genes. I can't remember how many I analysed for cubes. It would have been more than the 4 that metabolize. Oh, well, it's probably 1, 2, 3, 4, 5, 6. Probably 6 genes, 7 genes, analysed. And in this group here, which is Golden Teacher, Blue Magnolia, and Malabar, so the majority of cube cultivars, they all have the exact same alleles. They're all genetically identical at the psilocybin locus. The variation starts kicking in from luminous, so these little green dots scattered everywhere, penis envy, and in Australia. And what I've been doing for the last 6 months is starting to cross our 5 kind of different ways that cubes will produce psilocybin in Australia based on their genes. And I've been crossing them with everything that I know is different to see what will happen. And we've got a whole bunch of genotypes that are freeze-dried and just waiting for analysis under a bioplatforms grant. And hopefully one day I can share that knowledge with you. Subs. Now, if I tried to show the sub-diversity at the psilocybin locus in a haplotype network, so like what we did for the cubes just now, there would just be too many circles because there is so much variation. So I've used a SplitsTree, and pretty much in your domes, you can think about subs as differing among every single population, and even if your siblings are coming from a parental dikaryon, you might start differing at the psilocybin locus as well, because there is recombination at the locus itself. So there is extremely high allelic diversity to produce psilocybin in Psilocybe subaeruginosa in natural populations. And the million-dollar question, of course, is why? Why does it need to have such high allelic diversity? And for this, we're going to just have a quick little look-see at Tojima's D, which is that measure or sort of way to measure what type of selection a gene might be under, whether it's positive selection where everything ends up having that one allele, or balancing selection where there's often going to be multiple alleles, and there could be an evolutionary advantage to having multiple alleles in your population. And even though we haven't done this probably as well as we could have, we've got some evidence, some not convincing, of balancing selection. So, and I would lean towards saying that there is potentially, if someone was to really tease this out with excellent sampling in the future, there is potentially a benefit to having different alleles to produce psilocybin in the populations of Psilocybe subaeruginosa in their natural environment. So really, really interesting to me, and I'm super excited for the next person who starts to come along to tease out why there is such high allelic diversity at these psilocybin genes. And I'll just say subs are a great model to do this in, because we know their center of origin, we know they're here, they're easy to collect, and now we're pretty familiar with the locus itself. Okay, and this slide is about psiH. So a few talks back, I was talking about the role of psiH, its potential role in the cause of woodlovers paralysis. And so in that psilocybin pathway, the 4 genes that metabolize tryptophan into psilocin or psilocybin, you've got psi-D, psi-M, psi-H, and psi-K, which all help change tryptophan into psilocybin. I can't exactly remember what psiH does, but what's so interesting, it probably does 4-hydroxytryptamine into norbaeocystin, and because there's 3 copies of psiH, our hypothesis was, well, with all these additional copies of this similar gene, it's possible that they're modifying a tryptamine, and maybe that new tryptamine that's produced, instead of going towards psilocybin, it produces a tryptamine that maybe can bind to a peripheral serotonin receptor and cause a temporary paralysis during a psilocybin experience. And that's yet to be tested in any way, shape, or form. But what this analysis shows is the variability of psiH in these natural populations. So the first psiH, which we just call psiH1, is present in all of them as a functional gene, or a putatively functional gene. That's this clade here. So the other 2 copies of psiH, psiH2, sometimes has a functional or a putatively functional copy, and sometimes it doesn't. And psiH3 is almost completely a pseudogene, so it probably doesn't have a functional copy, and that's just based on like deleterious or like premature stop codons, and that that particular allele itself probably isn't transcribed or isn't making a protein. Occasionally, you get this switch of psiH3 and psiH2, so they lose synteny. So psiH2 is suddenly in the position that psiH3 would be. The take home of all of this is we've got heaps of variation at the psilocybin locus, and then at these interesting psiH genes where there's been a gene duplication, we still have a lot going on as well. So we've got genes that suddenly may or may not work, or that get switched around. And if they have a function in woodlover's paralysis, it's just going to be so interesting to know. Okay, and so what I've been doing, or for the last 2.5 years, I've been working for UQ, but now my contract finished in December, and I feel free to say that the reason I first started working on magic mushrooms is because I tried them many, many moons ago, and it pretty much set my trajectory into wanting to work on mycology. And then finally, after 16 years of studying mycology, I worked on magic mushrooms and have had this little adventure that's been so much fun to experience. And that whole time, of course, I have the same question as all of you, is what the hell is psilocybin doing in the wild? Or why is it here? And I am not very inclined to ever form opinions about things just because I don't know enough, but I've given this a lot of thought and have spent a lot of time now studying this allelic variation at the psilocybin locus, and I feel I finally have my thoughts clear enough to share them with you as to why I think that we're going to have a pathway toward designer shrooms. So to be on the same page as me, I'm just going to take you through a little bit about the background of psilocybin. So Psilocybe itself, under a clock-like rate of speciation, Psilocybe is probably 20 to 100 million years old, and that's just a kind of the mean age of genera in Basidiomycota, and it sounds like a massive age gap, and it is. So we maybe suggest it's toward the 90 rather the 100 million years than the 20 million years. And importantly, we think that psilocybin has been around that same length of time because everything that shared a most recent common ancestor with the ancestor of Psilocybe produces psilocybin, except for one species. So psilocybin's been around for a long time, and what we can be sure about is that it's designed to target serotonin receptors in metazoans. Metazoans is the most recent common ancestor of everything that moves that isn't a bacterium or in the sister group to plants, and of course archaea as well. So everything that has a serotonin receptor in metazoans, it could be mollusca like slugs, it could be insects, and it could be nematodes or something else that I haven't thought about, but things that might have been eating mushrooms 100 million years ago. And so because we know that fungi produce secondary metabolites to repel, to attract, or to take control, we know that psilocybin probably has a function like that. And over the last 100 million years, or however old Psilocybe is, we've seen this speciation to get to the point where now across the world we have Psilocybe in these shared niches, such as in dung and leaf litter or wood chips, or in mosses, these kind of shared ecological environments that Psilocybe thrives in, and it has this rich diversity. And so do a little thought experiment with me. Maybe you're a Psilocybe subaeruginosa and your primary predator is slugs, and maybe you're a Psilocybe in Central America and your primary predator is nematodes. And over 200 years, over the evolutionary time scale with a co-evolutionary arms race, what we've seen time and again is that secondary metabolites are honed for their targets. So if you've got a slug versus an insect or a nematode, it's possible that psilocybin and that pathway is being best honed through evolution to target its target, or to have an effect on its target, whatever that effect might be. So what I'm saying here is, if it's not too much of a stretch for you to join me on, imagine if different species of Psilocybe can cause a different psilocybin experience because of their evolutionary history and the reasons that they produce psilocybin for their target organisms that are eating them. So then the next leap that you have to take is that within one species, that allelic diversity that is there is also causing a difference in a psilocybin experience. And probably the easiest way to think about it is that if the psilocybin pathway causes woodlovers paralysis through different copies of alleles in that pathway, then it's really a chalk and cheese difference between two different mushrooms, one that causes woodlovers paralysis and one that doesn't. So that's an extreme example and it's still a hypothesis, but if you're with me that different genotypes of psilocybin can cause a different experience, then we're on the same page to design a shrooms. And for me to share that with you, it comes from when we first kind of started this journey together in this space. There was the dogma that it's psilocybin or psilocin binding to 5-hydroxy-2A tryptamine receptors, 5-HT2A receptors, and that's what causes a psilocybin experience. And then what you experience along that journey is based on that, and then set and setting. So now I'm saying, well, I think there's more to it than just the receptor and where you are and what you're listening to. So yeah, what I've been doing at Funky Fungus is starting to mix all these different alleles, and I think it's just going to be so cool if in the future, my dream is that you get to try different alleles and know that you're eating a different mushroom to everything else. I think in the community, with what we've seen with Kane and Simon's work on getting meaningful data for woodlovers paralysis, I don't think it's going to be that much of a stretch when we can start to articulate, and when we've got this baseline knowledge, this community can take us far in this question. And the knowledge that you need is that different populations of Psilocybe subaeruginosa are most likely different at their psilocybin genes. So if you're harvesting magic mushrooms in the upcoming season from different populations, from different wild sites, then keep them separate and see if you can notice a difference between sites. And of course, I don't advocate the use of magic mushrooms. Just going to say that. If you're collecting magic mushrooms from wood chips, in cultivated garden beds, then there's less chance that you're eating different alleles. Also, if you jump onto that cube paper and you get your hands on a copy, and you're someone who enjoys cultivating different varieties of cubes, well, now you can sort of say, well, hey, I know that this one was in the luminous group and it probably has a different psilocybin gene to this golden teacher that I've been growing, or this B positive that I've been growing my whole life. I want to see if there's a different experience and whether a cube is a cube. Won't it be fantastic when we find a way to start collecting these data and can make sense of whether there can be such a thing as designer shrooms? I personally think there can be. Okay, I'm going to skip over these outcomes because you already know what happened, and you were listening, no doubt. I put in this slide based on my last 6 months in this emerging psilocybin industry. And I think what I'd just like to say is that most of the knowledge that we have is based on the underground community. And that underground, Ronnie and I were talking about what the underground community means in terms of psilocybin. And underground maybe has a negative connotation, or it might suggest that it's a small community, and that's far from the truth. The underground community for magic mushrooms is enormous, and Ronnie came up with the analogy of an iceberg. So the underground community, which is forced into like this clandestine kind of communication because of the draconian laws against possessing or growing magic mushrooms and eating magic mushrooms, this community is this huge iceberg that's underneath the surface, and you are advancing knowledge on magic mushrooms. You've advanced knowledge on how to grow them, and I think we're going to advance even further. All the advances and all the knowledge that we have about magic mushrooms really stems from the community. I think the clinical use community, watching them since I've been in this space, is always interesting, but them just trying to get past the bottleneck of whether a trip is real or not compared to a negative control is, I think that this is a glacial pace. If the underground community is that iceberg, well, yeah, the psychiatrist is this glacier on top, going really, really slowly, and the vested interest in the actual cost of psychedelic assisted therapy to me is convincing that I love seeing the real advance in knowledge coming from the black and grey markets. We'll talk quickly about the vested interests here. So I'm in the natural production psilocybin, and that is something that you'll have to consider. So I have a vested interest. Then you've got synthetic production, and synthetic psilocybin can be produced for about $2,000 a gram. And biosynthetic production, which is producing psilocybin in E. coli or Saccharomyces cerevisiae, so like brewer's yeast or aspergillus, and that's under patent. And you can probably produce large amounts biosynthetically, and potentially you would get the landscape of tryptamines from a natural mushroom. So yeah, it'll be interesting to see how psilocybin ends up getting delivered to people who need it. And the last thing I'll just say is that the way that I've landed on how I will advocate for psilocybin in the future is that yes, 40% of people in their lifetimes will experience a mental health disorder, and psilocybin may benefit some of them. But what about the other 60% of the population who psilocybin can have a positive impact for? That's who I care about now, and that's who I want to start seeing getting the opportunity to take designer shrooms without being penalised by the law. So yeah, this has been so much fun, and it's been fun because of you all. Like I said at the start, 16 years working on fungi, and I think I was just passing through. Nobody really cared what I was doing, and it's just been so flattering that you've all taken such an interest in what I've been doing, and I really appreciate all the support along the way from anyone who's ever read or watched me speak about magic mushrooms. So thanks a milli, and bye for meow. Okay, actually not bye for meow. Ronnie has given me a couple of questions to answer that were given on the night, and I answered them. But now, this is actually a couple of days later, and I've re-recorded my talk because it was so, the audio was so terrible. And yeah, these are the questions that were asked at the end of the talk. So how far can spores spread themselves naturally? Now, mushrooms produce copious amounts of spores, huge amounts, 2,000 spores per minute. And these go on the whim of the breeze to wherever they can make it to, as far as they can. And when we're talking about mountaintop populations or populations within valleys, so I'm thinking of Psilocybe subaeruginosa, obviously there's a limit to how spores can travel, because they might reach barriers, whether it's a geographical barrier like a desert and spores don't survive, or a bunch of trees that they just can't get past. And we've seen this with Psilocybe subaeruginosa, that there probably isn't contemporary gene flow between a lot of these populations. So there are limits to how far spores can spread. And when we think of pan-mictic populations, which is animals are pan-mictic because they can move across barriers, no wuckers, whereas mushrooms, you can have these pan-mictic populations that spread far and wide, probably through spores. It just depends on the geography, I think. And I think as people look at more at population biology and mushrooms, they're starting to see that there are geographic barriers or there are localized populations. The next question, which I think was from someone called Graham, and I love it, is sexual compatibility the main benchmark used to describe a species? And on the night, I think I said something along the lines of, what is a species is often a question that professors ask their PhD students and whatnot, and there isn't necessarily a clean answer. Because life is so complex. So what's a species of a microorganism compared to a species of mammal, for instance? It's hard to come up with one answer. So the way that I am thinking about mushrooms at the moment is, and the way that I encourage you to think about mushrooms is, the species is kind of that overarching taxonomic rank, and then you've got populations, and those populations might be separated. They've all come from a most recent common ancestor, and they've just been separated for a while in a valley or on a mountaintop. And technically, if they ever had the opportunity to meet up again, they could cross. And we've seen that with Bunya in Psilocybe subaeruginosa crossing with Psilocybe subaeruginosa from Tasmania. So sexual compatibility is pretty neat. The jam, where you kind of run into a bit of a problem here, is that because these tetrapolar fungi, like subs and cubes, they benefit from differing at mating loci, so at having different alleles at their mating genes. It's kind of weird. Maybe they've been separated over space and time for a long period of time, but there hasn't been a loss of mating compatibility. It hasn't been under selection. So maybe they've speciated, but they can still cross. And that's what drives people wild, I guess, in kind of defining a species boundary based on a sexual kind of reproduction criterion with mushrooms or with fungi. Often we'll see people using large numbers of genes to describe a species, but yeah, geography, sexual compatibility, morphology, all of these of course can be considered, and you're kind of painting a picture. And in the future, I think population biology is going to become so much more relevant, and it's already very relevant. It's going to become so much more popular to show the boundaries of species. The next question is, have you seen sporeless Psilocybe cubensis in the wild? And I have not personally, and what I think I said on the night was something along the lines of, the reason that you wouldn't expect to is because it's a disadvantage to the mushroom itself. That genotype will not be able to produce spores to spread its alleles into future generations. So cubes most likely have ephemeral genotypes. We could think of them as annual plants, where they've been eaten, a spore has been eaten on a piece of grass by a cow, it's germinated in a nitrogen-rich piece of manure, met up with another spore, probably in the same piece of manure, colonized that manure with a dikaryotic genotype, and then fruited a mushroom. And then that mushroom will disappear, that genotype will disappear once that manure disappears. So it's an evolutionary dead end to not produce spores, and we see it in cultivation with these ones that are cultivated, where they no longer produce spores. It could be a mutation in a sporogenesis gene or something to do in the mating pathway where they've lost the ability to produce spores. In the wild, it's less of a desirable trait, but Kane has more knowledge about this, and I think, Ronnie, you got to stitch Kane in after this. Okay, and then Darklight, thank you Darklight, asked the question of what is the evolutionary purpose of psilocybin and what's going to be the next arms race for selection at the psilocybin locus. And of course, my answer as always is, man, I wish I knew. I wish we could go back that kind of 20 to 100 million years in time and look at who was eating psilocybe mushrooms and say, oh, okay, it's definitely this. It's definitely to interfere with their light inference, and they're going to start eating mushrooms during the day, and then their predators are going to eat them. Or it is taking control of their minds, and they're going off and somehow spreading spores of my mushrooms. Or it's just a straight up poison and things just don't feel so great and they're a bit disoriented and they know not to take another bite of me. Whatever that evolutionary purpose is, we can talk about the next arms race, and that comes from humans innovating psilocybin mushrooms for our own purposes and especially in the case of designer shrooms. So if genetic differences at the psilocybin locus do translate to different experiences or can be tailored for different disorders, such as one particular experience might be good for people who have anxiety, or one might be good under palliative care. Who knows? Because maybe it will just make you come to terms with death a little bit easier than one that's just having a shiny experience, or you're seeing cartoons everywhere, or it's excellent for music, or they all are that. Who knows? But I think humans are going to innovate the next arms race, and we're going to start seeing some special stuff, in my opinion. And do I even dare say anecdotally? Who knows? Will natural psilocybin be used clinically? And then kind of mixed in with the same question is, will extracts from magic mushrooms ever will ever fall under clinical use? So I really hope so, that natural psilocybin will be used clinically. And you saw on about 2 slides ago the different ways that you produce psilocybin: synthetically, biosynthetically, or from magic mushrooms themselves. And all the use so far has been from synthetically produced psilocybin. And it's interesting to see the impact of this in studies that have shown that there is no difference between somebody taking lysergic acid, psilocybin, and mescaline. And I do wonder if that is because they've used synthetic psilocybin in these trials, that the patients can't distinguish between these different trip experiences. Because I have heard that taking LSD is very different to taking a magic mushroom. And hopefully this is going to be a question answered by clinicians in the future, especially if they start looking at as well, and they can link it to the allelic diversity of psilocybin in magic mushrooms. So I hope it's going to become a direction of research in humans in the future. Obviously, it's already super hard just to get permission to work on psilocybin to begin with. So I think it's going to be a matter of time before we see this question answered, and which is also why I think it's best not answered in the clinical space. Now, using magic mushroom extracts, I think, this is a great way to produce medicine. So the problem is that it's probably harder to get past something like the TGA, the Therapeutic Goods Administration. So when you produce psilocybin synthetically, they know that there's one thing in whatever the mode of ingestion is, whether it's a tablet or you're somehow doing a psilocybin suppository, or you're doing a vape or whatever. So theoretically, what you would do is grow up one genotype of magic mushroom, and you would know that it's consistent over time and space because you're growing up a clone. And you would know that in 1 gram of that magic mushroom, you have a consistent production of psilocybin and the other tryptamines. And then you would either aim to just extract psilocybin, which I think would be very difficult because those tryptamines are very, very similar, or you would just extract the entire tryptamine landscape and give it as a natural magic mushroom experience extracted from a magic mushroom. I think the way that we were going at Funky is probably similar to this, which was to probably grind up consistent genotypes of magic mushroom to put directly into chocolate or gummies or whatever. The last question is, what are my hopes for the future in this space? And if you've watched the entire night, then you would have seen Simon answer this question really, really well, and I agree with him that I hope that that people who have done the research and who are aware of the risks of psilocybin could try a magic mushroom without facing legal penalty, and see if the experience benefits them for whatever reason they want to do it, or even if they just want to have a good time. My hope is that one day we all get to try these different alleles of the psilocybin pathway, and we can link them to different trip experiences. But that's my agenda. So I just hope that you can all experience different genotypes of magic mushroom in the future. Thanks for listening.